Molecular markers are heritable DNA variations that allow individuals, genotypes, or species to be distinguished. Many marker types correspond directly to sequence polymorphisms, including SNPs, substitutions, indels, repeat-based variants like microsatellites and minisatellites, as well as structural variations such as inversions, copy number variants, and translocations. Because these variants represent defined DNA changes at known genomic sites, they are considered non-anonymous or targeted markers. They assay a specific identifiable locus, and in many applications, particularly association studies and marker-assisted selection, such markers are intentionally chosen because they fall within genes or lie closely linked to functionally important loci, although they can occur anywhere in the genome. Detecting them requires methods tailored to the type of sequence change involved. SNPs are commonly identified by sequencing allele-specific PCR or SNP arrays, indels by PCR with fragment length analysis or sequencing, repeat-based markers by PCR and size separation, and larger structural variants by long-read sequencing, karyotyping, or FISH. Some widely used marker systems, RFLPs, AFLPs, and RAPDs, do not directly assay a known SNP, indel, or repeat. Instead, they function as techniques that generate sets of markers, typically in the form of fragment or banding patterns. Because these patterns can arise from many possible underlying sequence differences, the method itself, rather than a specific variant, defines the marker. RFLPs detect changes in restriction size or fragment lengths using restriction enzymes and electrophoresis, and because a locus-specific probe is used, these markers are also considered non-anonymous. AFLP and RAPD, by contrast, amplify many genomic fragments using selective or arbitrary primers. And because the chromosomal origin and causal sequence change for each band are not readily identifiable, these are considered anonymous markers. Another distinction among marker types is whether they produce codominant or dominant data. Codominant markers, including SNPs, microsatellites, indels, and most RFLPs, allow both alleles of a heterozygote to be distinguished, supporting accurate genotype calls, parental assignments, and detailed population genetic analysis. Dominant markers, such as AFLPs and RAPDs, score only the presence or absence of a band, preventing the separation of heterozygotes from dominant homozygotes. Although less informative, dominant systems are inexpensive and require no prior sequence knowledge. Marker systems also differ in abundance and informativeness. SNPs are extremely common but usually biallelic, whereas microsatellites and minisatellites are less frequent yet highly polymorphic. Structural variants are rarer but often informative across large genomic regions because they span thousands to millions of base pairs. Identifying such a variant yields information not only about the mutation itself, but also about the surrounding chromosomal segment, including gene order, haplotype structure, and regions of reduced recombination. Taken together, these features determine which type of molecular marker is most suitable for specific applications, including evolutionary studies, breeding programs, and genome-wide analysis. If you found this video useful, Do have a look at the other videos on this channel, hit the like button, and support the channel by subscribing.